Hi everyone, welcome back to Animal Behavior Live 2025. I'm Chung, and I'm thrilled to be chairing this session. We're kicking off the first section of the day with a fantastic speaker, Dr. Wen-Sung, a senior research fellow in the Marine Sensory Ecology Group at the University of Queensland, Australia. Dr. Chung's team blends high-resolution MRI with behavioral research to reveal neural circuit, neural architecture underlying cephalopod intelligence. Truly some of the most fascinating work happening in the field at the moment. Some of you might already know Wen-Sung from our Meet the PI podcast episode, where he shared not only his scientific journey, but also a bit about life beyond academia. If you haven't listened yet, I highly recommend you to check it out. Today, Wen-Sung will take us into the world of cephalopods, exploring how sexual dimorphism and neuroplasticity shape both brain structure and behavior. Before we dive in, a quick reminder that please post your question in the YouTube chat. Wen-Sung will answer them during the Q&A section. And now, without keeping you waiting for too long, Wen-Sung, the floor is yours.
Thank you for the wonderful introduction, and it's a grateful to have this opportunity to share my study with you guys. So today I'm going to talk about my journey to working on one of the very large brain invertebrate, cephalopods. So today we're specifically talking about octopus. Before jumping into my talk, I want to acknowledge the Quantum people as the traditional owner of the land and the sea which I conduct my research. And because their permission and the knowledge, I can working on some very iconic Australian species. I respect their elder, past, present, and emerging. So before I want to introduce our group, it's a Marine Sensory Ecology group at UQ. We're working on several different animal groups, including fish, stomatopods, the mantis shrimp, and the cephalopods. So Justin was our big boss, retired a couple of years ago. Now Fabio and Karen is working on fish, and my study is more focused on cephalopods. As you can see, we are working on several different animal groups, so we can do a lot of interesting study, also enjoy a lot of field work. Our field work basically focus along Queensland coast. So especially we spend a lot of time in this island research station, also two UQ Marine Station here on island and the Moreton Bay Research Station. For my personal interest, I spend a lot of time in Moreton Bay Research Station because that area I can catch many different species for my study. So here we can have a quick look of our wintertime office in Lees Island. You can see the beautiful ocean, the lagoon, and then we did a lot of diving over there. You can see the habitat is still very healthy. And we also have the compulsory beach time to drink beer every afternoon. So what is cephalopods? They are kraken. They also the fishing bait and calamari for the food, and a classical model for neuroscience, especially back to 50s. The giant axon used to be the very important neuroscience material to figure out the neural signal transduction. Also, octopus can predict a World Cup who will win the game. Cephalopod actually belong to mollusca, but it's more advanced because they get rid of the external shell and make the body shape is more hydrodynamic, make them swim very fast. They also have a very advanced sensory system, allowing them to do very efficient hunting in the marine environment. So I call them racing snail. Their lifespan very short, and they also have 3 hearts, blue blood, very large brain, very smart behavior, And most often only live for 1 year. You can see here is the octopus mom looking after the eggs, and this the hatchling direct blew out by the octopus mom through the funnel. And here is a 1-day-old blue line octopus. This one is 1 month old. Another thing I want to emphasize, the diversity across the global is very different. So you can see in the Indo-Pacific region, we have a much higher diversity from Japan, Taiwan, Philippines, and Australia, especially octopus and cuttlefish. And the one interesting fact is in America, you cannot find cuttlefish. There's no cuttlefish living in American waters. How do we working on this animal? We using different types sampling method. So in the reef, we're using a small boat, doing a lot of scuba diving. And we're also working in the open ocean using super yacht, which is occasionally we have opportunity using submarine to dive down to 1,000 meters to collect deep sea specimen. About 10 years ago, I using this submarine to film the first ever live giant squid footage in their natural habitat in Japan. We're also using the massive research vessel. Here is, you can see that's me, and using this German research vessel, we collect a lot of deep sea specimen. So in case you haven't seen different types deep sea cephalopod, here is some example. Can you recognize the fresh live deep sea specimen and match with this museum specimen? Give you 5 seconds. Can you match which one belong to which one before, after preservation? So you can see their morphology change a lot. So it's very difficult to match their live body form compared to a museum sample until you really spend time in the field to observe their behavior and what they really look like in their daily life. So I was super lucky. So I have a lot of opportunity to see this animal in their natural habitat in different depths. As I mentioned, they belong to mollusk, but they are very different. Compared to a normal mollusk, they have very simple nervous system. Usually around, they only have approximately 20,000 neurons. However, cephalopod become more complex. They have many more neurons, up to 500 million neurons, and their brain is more specialized for different specific lobes for different functions. For instance, they have a very large optic lobe. Here is in charge for the vision task, and then they also have the vertical lobe. It's like a cortex for learning memory purpose. Lucky enough, a couple years ago in the deep sea cruise, I caught this almost transparent octopus. So this gives us an opportunity to see their nervous system. So you can see here, it's a central brain. They have a very weird looking telescope eye. And then you can see ganglia, so along their arms. Every single sucker have one ganglion in charge of those local image processing, information processing, including sucker control, muscle control, and the chemotactile. However, not every octopus is like this transparent one, so we need a special tool to study their nervous system. And you can see they have very large central brain. Why they need such big brain for this supercharged snail? One reason is they have the very complex skin. So on their skin, they have a lot of chromatophores. Every single chromatophore can be controlled by their brain to change the patterns. So you can see this cuttlefish showing this passing cloud pattern to attract the female sitting behind the camera. So they can use this display panel to change their body pattern for camouflage, for signal communication, and even for some very complex behavior. I will show you later. So one very important function is camouflage. You can see this reef algae octopus from this body pattern to become camouflage just within 1 second. here and there, and here almost disappear. So next time when you have opportunity in the ocean, you might pay more attention. Maybe a lot of octopus just sitting around, but where the camouflage is just unable to see by your human eye. Now I give you a challenge. Can you find 3 octopuses within 10 seconds? So this guy are very active during daytime in the reef. Now you can see 3 of them. The arrowhead point to their eyeball. Not just the color match, they also using the skin to change the structure to match the background. So they are very good in reef habitat and also give us a very challenged time to find these well camouflage octopuses. Not just for camouflage, they also using body pattern for communication. Here showing the male octopus using the black stripe, try to attract the female. However, this female just not interesting about his pattern. So showing the refuse body pattern here. So aside from the camouflage and all those color change capability, they also perform a lot of smart behavior. And this also inspire Hollywood to produce a movie and a documentary. And what they really can do. So some of them will use a tool like a coconut shell. The octopus will collect this as the shelter. And also they are very smart to learn new tricks. So if you give an octopus a camera, he can figure out how to do the selfie within 1 day. So just like my daughter when she was 2 years old. I also train octopus to do some tasks. So this one is need to learn to pick the circle and get the reward. So you can see octopus is looking and attack the circle sign and get the reward. And on the right-hand side, you can see in the naive one, they will spend more time to explore both sides. And then after a couple of trial, they become much efficient to make the decision. However, octopus also have very strong personality. So it's sometimes they learn very different way. So like this one, he learned how to do this task, but after 10 trials and refused to do any task when I gave him. Because he figured out even he didn't do the task, he will still get the reward eventually. So it's just like my son, when he insisted want to have the pizza and I would refuse to eat other dinner. Also, I train my octopus to do the counting task. So the octopus need to choose the big number versus the small number. So for instance, this one need to choose 2 balls versus 1 and I get the reward. So you can see attack the 2 balls and get a reward. And then we do more difficult challenge is like a 4 versus 3 or 2 versus 1, 3 versus 1, and see how far they can go. So far, no octopus can pass 9 balls task because they only have 8 arms. I want to show you one classical study that's a very early study done by John Messenger. So it's a cuttlefish learn to attack fish in the jar. All right. So the normal cuttlefish will learn they unable to get fish because fish trapped in the jar and give up to trial for the next time. So just once they know, oh, it's unable to get the fish, they will stop doing this attack. However, if J.J. Yang actually removed surgery, using surgery to remove a part of brain, and then he figured out the cuttlefish unable to remember what he just learned. So remove the vertical lobe actually will stop the learning, they will continue to attack the fish in the jar. So this is the very first time to demonstrate using the octopus or cephalopod in general, using the certain brain region for learning memory task. J.J. Yang continued working on this experiment using octopus and figure out if we remove the optic lobe, then unable to learn any visual learning task. If remove the vertical lobe, in general, they will fail all those learning tasks. If remove the inferior frontal lobe, then all those chemotactile learning tasks unable to be remember. But remember, all those invasive study is back to 50s, 60s. In this day, this kind of invasive study become very difficult to perform because the animal ethics requirement. And that is also not so human to do this very intelligent animal. But because those classical study also inspire us, because this is a very different types group of animal, they actually have very advanced cognition behavior compared like a COVID or the chimpanzee, but they in the phylogeny they locate in the very different phylogeny group, and this also give us to reconsider why cognition develop in different animal group. So a lot of the challenge for study cephalopods neuroscience and cognition. The one challenge is most of our knowledge still based on a few species in Europe, especially the octopus vagaris, the common European octopus. So J.Z. Young basically using this species as the model animal to do a lot of neuroscience studies and the behavior approach. So it's become like a neuroscience, octopus neuroscience Bible based on J.Z. Young study. And also very difficult to extend the complex brain because the brain become too big, too complex, and it's very difficult using the conventional histology to study this complex neural system. Also, small sample size and the different types of labor condition, and sometimes we just unable to repeat the study. And also, we have very limited knowledge about their phylogeny relationship and the variation during ontogeny and sexual dimorphism, all because the challenge I just mentioned before. So consider recently we have many different types of new technology can investigate this fascinating animal group. Especially at UQ, we have the Advanced Image Center. We have many different types of MRI machine. So that's the beginning of my journey using this non-invasive way to study cephalopod brain and the nervous system. So far, I have scanned many different species. In the beginning, just because I want to study those very rare deep sea specimen. So like this one is a telescope eye octopus, and this the vampire squid is super rare, and only available. We only can catch them below 1 kilometer depth water. So because they are so difficult to get, so I want to use the non-invasive way to figure out the strategy before I cut them open. So here you can see I already studied many different groups, including nautilus, many types squid, like this super cute pajama squid, and then many types octopus. Paper nautilus is also a kind of a pelagic octopus, and some deep sea we're looking specimen. Like this one is the strawberry squid. This one has their left eyeball usually 3 times bigger than right eye. So we also put this specimen into the MRI scanner to see the internal structure. So here is give you some idea. We put a fixed animal into the high resolution scanner to image the whole specimen. So you can see here, we can see the internal organ, all those detail from the eyeball, brain, and the digestive system, and the venom, and the neural tract. So that's the very beginning process, and then we want to get more high resolution and get into the neural circuit. So I will tell you in later. So in the beginning, I just scanned many different species across from different depths, including this all from the deep water between 200 down to 1,500 meters. You can see they have massive eyeball because living in the dark environment, this visual predator, they rely on their vision mainly, so they have a lot of adaptation in their visual system. And compared to octopus, you can see their eyeball design is very different compared to squid or cuttlefish. And in general, all of them have a very large eyeball and optic lobe. That's also emphasize these guys depend on their vision a lot for their daily task. And here, give you some idea, their central brain, you can see the morphology is quite different. The octopus have the most compact nervous system compared to squid because they're streamlined body shape, so they also have a more elongated brain design. So we also can use this morphology to identify the different species, the different group based on their brain design, outlet, layout. Although we can get very high resolution of the whole animal scan, but you can see the brain resolution is not so perfect. We can see the overall structure, but no detail in more fine structure. So we developed a different approach. We basically isolate the brain, and you can see here we push the resolution very close to the histology resolution now, using different contrast agent and optimize our scanning protocol. So you can see now the structure is so clear. We can see all those detailed structure within the brain. After I got this tool, now I want to ask a question. If all octopus or cephalopod in general, they have a very similar brain design, or do they have a very different brain design? Depends on where they live or what kind daily task they encounter. So I choose 4 different octopuses from different depths. So the first one is the vampire squid, live below 1,000 meters. And the second one is the blue line octopus. It's a very iconic Australian species because the reputation of their venom. So they have a very strong TTX, and usually for the defense or the hunting. So they can easily to paralyze the prey, even human. So there's a few incidents because the human put the blue line octopus in under their wetsuit and beaten by this guy, and caused very bad incident for that human. And then we can compare from different habitats, different this one is nocturnal one, and these 2 are diurnal octopus. They are very active during nighttime, during daytime only in the reef. So we can compare different behavior and see if their brain have a different development for their spatial life. So here is the vampire squid, the deep sea one. You can see the brain become very simple, very smooth. I guess because they live in the deep water, so not necessarily doing a lot of complex tasks. So it's more like a koala. They spend a lot of time just hugging the tree and sitting on the tree. So they don't really need a very complex brain for their daily task. But compared to those reef octopus, they do have very different brain design. So you can see this have a brain folding on 2 reef octopus compared to the normal octopus. They have more simple brain structure. So one thing you can see here is their optic lobe. All of them have a very, very large optic lobe. So also emphasize the vision is very important for this animal. Although they are colorblind, but they do have a capability to do a lot of visual navigation, visual communication, and also hunting, all those tasks. So here you can see their brain design is actually quite different between nocturnal and the diurnal species. The first thing we see the optic lobe in the nocturnal species is relatively small compared to diurnal one. But during the nighttime, not so much light, so they rely on more chemosensory. You can see the brain the chemosensory region is much obvious, more dominant compared to the daytime octopus. So, well, because the morphology, so we want to ask, do they really have the different function? So we're using electrophysiology to measure their vision. So nocturnal octopus, they colorblind, only can see the blue-green color, very similar like their sibling live in the reef during active during daytime. They both only can see blue or green. However, one thing very different is they can detect very different the fast objects for the day reef octopus. They can follow very fast flicker frequency. This is the first time we demonstrate the invertebrate can see very fast flicker signal. Also indicate they can track those moving fast objects in their natural habitat. So give you some idea, human eye only can see roughly 60 hertz of flickering. So anything faster than that, we was unable to follow. So you can see the reef octopus actually do very good task, very good vision to live in that very complex habitat. Another interesting discovery is their vertical lobe. As I mentioned before, vertical lobe is for learning and memory. It's like a vertebrate cortex. And we found they have folding. It's like a wrinkle of the vertebrate brain. Now, this is the very first time we found in the invertebrates also have this kind of brain structure to help them to have more specific subdivision for different tasks. So you can see deep sea species with one lobe is very smooth. Like I mentioned in koala, they have a very smooth brain. But the normal octopus, nocturnal solitary octopus, they have 5 subdivisions. Compared to reef octopus, those very active during daytime, they have additional subdivision, so it have 7, not just in the vertical lobe, also in the optic lobe. So for the normal octopus, they only got like a bean-shaped optic lobe, but the reef octopus actually have modification of their optic lobe into more complex shape, look more like a croissant, and they have additional folding to allow more signal input in order to deal with a very complex visual scene. So here is an example of reef octopus. It not just doing awesome camouflage tricks, also have a very special behavior. They have a cooperation hunting behavior with the fish. So you can see the coral trout, when it come in, it's actually gray, but now it changed the color to the brown patch pattern. It's actually tell octopus, oh, that's hunting together. So they will swim together and find prey, and the coral trout will do the handstanding posture to point out where is the little crab or shrimp hiding in the crevice or below coral. So octopus were using his arm crown and using 8 arms to tickle those little prey out from the crevice. And some flush out prey will be eaten by the coral trout. So they have cooperation for this hunting trip. One interesting question here is octopus only live for 1 year, but coral trout can live up to 20 years. So coral trout might maybe learn this cooperation from their siblings, from their neighbors, but octopus need to learn from the beginning. So because they live very short time, so they need to learn this very quickly in order to survive in that habitat. So not just their ecology and the living mode will affect their brain design, also match with their phylogeny. So you can see here is a vampire juices, it's a deep sea octopus with a very smooth brain, and the reef octopus actually become diurnal and very only active during daytime, and have more brain folding in order to deal with that very complex visual task compared to nocturnal one. They have a smooth optic lobe, but 5 subdivision of the vertical lobe. So this interesting result to tell us actually octopus brain is evolved based on their daily task and also match with their phylogeny relationship. So let's have a look of why vertebrate brain have this brain folding. Maybe because they need to match with their ecological benefits and the social benefits. So including all those different benefits, because they need lots of brain power to deal with all these very difficult behavior tasks. So potentially, octopus also encounter this challenge, and these guys also have the brain folding design in order to deal with the like a foraging task, home range finding, also pair bonding, social learning, for instance. Okay, and then I'm jumping into the next field. Recently, I more get into the sexual dimorphism because this very common feature in the nature. You can see so many different types sexual dimorphism from the body pattern, color pattern, body size, behavior. So they are really fascinating. Like paradise birds and butterflies, and even the jumping spider. You can see this Queensland peacock jumping spider. The male is so beautiful and have a fascinating dancing to attract female. And I wonder if in cephalopod also have a very similar adaptation in their daily life. Okay, so you can see, yes, cuttlefish also iconic example. So this is the Australian giant cuttlefish. You can see the male is become very large and the male is relatively small. And the male can display so many different body patterns to attract the female. And then a few more example is like octopus. The male have the enlarged sucker. It's like a sexual feature to attract the female with this very special enlarged sucker. And then another extreme case in octopus is the dwarf male, like this Pepernotheus. You can see the male is become tiny. And then a few more examples like this 7-arm deep water octopus. You can see the body size change so much. Recently, we understand cephalopod actually using different types of sex chromosome, like the coleoid octopus, cephalopod, including octopus, cuttlefish, and squid. They have this double Z system for the male, and the Z0 is for female. But Nautilus using the normal XY and XX sex chromosome. So all this just new information and make me more interested in if the sexual dimorphism will affect this animal group. So I'm particularly interested in octopuses. So you can see here the species I'm really interested in. So this is the Pepernotheus argonauta. This is the female. The female will make the shell as the egg case to brew the babies. The male is tiny, so usually just attach their mating arm on the female body and die. And then this one is the blue octopus. The female usually much bigger, and a few other deep sea species. Because the time limitation, so now I will just focus on blue line octopus. So blue line octopus, the reputation about their neurotoxin, because it is so powerful, so it can actually paralyze people if you're bitten by this animal. You roughly have 5 minutes to ask someone to help you or to call ambulance to rescue you. So you can see here we found this Queensland blue line octopus. The female is usually much bigger than male. And then here you can see the behavior. Because they have short life, very short lifespan, and then when they get mature, the female ready for spawning will become very hungry and want to eat everything in order to deal with the very long period of the brooding time. So female actually will eat everything she can catch. So in this case, it will give male octopus high risk because sexual cannibalism will occur very often. But if this is a real case, that we can imagine the male blue line octopus probably will extinct in the field. And what they can do to offset this risk, then that's become a very interesting story now. Okay. As I mentioned, TTX is so effective, so even just small amount is enough to paralyze prey. We found the small male blue line octopus actually have very large venom gland. What's going on? So you can see here is the mating behavior. You can see the little male approach to female. And the mounting on top of the female. So on this corner, you can see the breathing rate. The male become very exciting, and because he need to holding the female, you can see the ventilation rate is much higher, nearly double compared to the rest time. Interestingly, the female breathing rate is gradually drop. So you can see here the small male sit on top of the females. At this stage, the female still can control the body pattern, and we gradually see the breathing rate drop almost to zero within 5 minutes. After 5 minutes, you can see female lose the muscle control and it become pale, and lose all those reflex. Even with the very strong flashlight, the pupil lose reaction. So during this period, female stop breathing for the whole mating period for nearly 1 hour. So after 1 hour, you can see female gradually get the muscle control back and push the male away. Then you can see a hump on top of the female body, usually nearby the head or the mantle. So this big hump is actually the biting side. The male pump TTX to female body to calm her down, to avoid being eaten by the female. So you can see here the different biting sites. So you can see this little hump. And we are using the MRI to figure out why male is choose the certain biting site. Then you can see these are 3 hearts and the aorta. The male, for some reason, can aim for this region to make the TTS get into female body more efficiently. So using this way, the male can finish the mating behavior and avoid being eaten by the female. And I need to emphasize, no female have any incident by this TTX injection by the male. So that's the natural arm race between a male and female. So that's the special way this animal evolve, have this special mating behavior. Because I'm interested in the brand, and I scan many different individuals from different life stages, male, females. So one very big challenge is because this guy, they don't have a heart structure to keep their brain intact. So even handling capture or fixation will cause the morphology distortion. So this give us a big headache. So you can see here, 10 different individuals, all of them look different. So it's make the brain analysis it become very challenged because everyone will have the difficulty to identify the different part of the brain region. So I'm thinking about, do we have any solution like a human brain scan, have a brain templates like this one? It's based on 152 people brain MRI scan and then make average brain templates. And all our current study, we can use that as the standard to compare like during ontogeny in different age or like here. So you can see male, female have a brain development differences and in different ages. Yeah, so that's become my dream. Want to figure out if we have a solution to make octopus brain study can adapt this method. So this become a long journey because their brain is more complicated and it's very different compared to the model animals. So we start this journey from last year. So in the beginning, we just try to align this brain together. You can see it's unable to match because the morphology deformation. So how to make this become a beautiful brand template is become image analysis challenge. But lucky enough, we have a lot of expert at UQ, and with those mouse brain fundamental study, we actually have a lot of tool can do this task. So here, give you some idea. This is the brand template we want to approach, and then list the different types of brand. And using the image processing, we can register brand, different individual brand into the brand template, and then keep all those variation like this one. You can see it needs some stretch or compress to make the brand to allow the brand to match with the template. Also keep all those information for afterward analysis. So here the red region indicates the brand actually expanding to match with the templates. The blue is a compress. So all those information still keep in the matrix. We can use those information for further analyze. So here's the first ever octopus brand template. You can see it's become very clear. All those boundaries become so clear that allow me to do the segmentation in the very high detail. So now we can separate all those brands into 51 different functional brand regions. And then we can use this segmentation reduce back to every single brand. So it saves a lot of time to do the manual segmentation. So one very interesting discovery is we can see here, we know they have a sexual dimorphism in their body size. Female is much bigger compared to male. Okay, you can see here. As I also mentioned, female will spend roughly 50 days for look after eggs. So you can see they lose a lot of body weight because during that time, females stop eating, but just looking after eggs. So you can see the body weight is changed. Interestingly, their brain is very different. You can see the female brain is actually much smaller compared to males, although males have a smaller body. So what is going on this iconic the blueies? Yeah. So we found the male has much bigger optic lobe, so it's a visual center. Compared to female, the optic lobe is relatively small. So one hypothesis is because female, when she ready for spawning, she usually stay in the shell, just ready for spawning and look after eggs. So at that time, the vision probably not so important. So that's probably one reason they don't invest so much energy in keeping a very large visual center. But they keep their central complex in charge, like arm control to look after eggs. So using the template, we can now do the Jacobian analysis. So here, the blue optic lobe indicates the male optic lobe is much bigger compared to female. And then the orange part is the optic gland. Optic gland is like a pituitary gland in charge the whole growth hormone and the reproduction. So at this stage, because the female is ready for spawning, so the optic lobe is still keeping growing, even overall female brain start shrink. But this gland also produce the suicidal hormone. So when this process kick in, the female actually start their process, start digest herself to transfer all those energy to keep her look after eggs through those very long brooding periods. So you can see here. Okay. One thing I need to emphasize here, although female brain start shrinking, but female always keep their chemotactile brain region intact, even to the end of the life, for one reason. The female need to look after eggs. You can see using the suckers to detect, touch eggs to make sure to extend embryo health. What if unhealthy embryo detected by the female? Then you can see this footage. So female actually can push or throw the unhealthy embryo away. So in this way, female can ensure all those clutch in the good condition, make sure the very high success rate for the hatchling. For one of my students actually doing more detailed behavior assays, so we test how good the female can recognize the eggs. So we steal her eggs and put it back. Female immediately recognize that her own brood. And then we also swap different females' eggs, and then she still can recognize this belong to me and those from the foreign eggs, and will throw that away eventually. So this is the very first time we demonstrate octopus actually have the blue awareness, can recognize her own eggs. So aside from the morphology, the brain morphology, we want to see more detail. So we're using different MRI technique. It's called diffusion MRI. Using this way, we can detect the neural tract within the brain. So we can use this technology to detect the neural network within the brain. You can see the blue indicate the nerve is going from left to right. The green is from anterior to posterior. The red is from dorsal to ventral. So using this way, we can examine the neural circuit and reconstruct the connectome. So here showing one example. If you're using histology way to reconstruct the motor neuron, you'll probably take whole year to reconstruct all those 13 different pairs motor neurons. But using diffusion MRI, we can do this task within a week. And it give us much better visualization, 3D view, so to track how they go and how they connect with others. So we also can do the connectome. So as I mentioned, we can separate the brain into 51 different functional regions. And we also can detect the connection strengths between different lobes. Here, showing the mature female before spawning, you can see all those connection strengths. The blue indicates no connection. The red indicates a very strong connection between different lobes. And then compared to this senescent female is close to the end of her life, you can see a lot of region the connection disappeared. So not just the brain shrinkage, they also lose the connection. So this help us to figure out what is going on, why female getting old and lose the control. For instance, this female unable to control her arm anymore because all those connection circuit is actually disappeared. So using this way, we can see from the morphology and then down to neural circuit level. And then not just doing the same species, the male-female comparison. Using this brain template, we actually can do the similar image processing to reduce this reef octopus into the brain template. You can see the brain is actually very can match with the templates and do like a Jacobian analysis to see how much difference between two different species. And this opened a new field. So a lot of potential application, like we can reduce the image from the micro-CT or the advanced microscopy, like the light sheet or volume EM into this MI-based brain structure templates, and then doing male-female comparison before-after learning task or species A versus B, young versus the old, or the different environment change. So this gives us a new way to study octopus brain or any cephalopod brain. So this will open a new way to help us understand this complex animal and how they evolve into the current complex brain structure. Not just doing the gross anatomy, we also involve a lot of work with the Cambridge. So doing the ultra high resolution microcity and the EN, we try to figure out the synaptic connectome. So in this stage, we can our study now we can investigate evolution process or the individual variants, ontogeny, and even get into the neural connectome, the synaptic connectome. So we try to investigate in different levels. And then our future project is we want to connect to the molecular work. So not just the connectome, also the single cell sequencing, and figure out the neural network, and potentially we can apply this new knowledge into the robot design. So here a few take-home message. So evolutionary help this ancient animal develop a certain complex structure in their brain and their behavior in life. And depends on where they live, they have a different adaptation either for the vision or for the chemosensory. And the octopus brain template is a good toolkit for many different applications in the future. And I want to thank you for my small octopus team to look after our octopuses and our colleague in the sensory group and the founding source. And I'm happy to take questions. Thank you.
Right. Thank you for the very exciting talk, Wen-Sung. We only have a question from the audience. The first one is from David. Are you able to scan live specimens or only that one? And can you use preserved museum specimens or do they have to be freshly caught?
Okay. At this stage, we're unable to scan live animal yet because we try to figure out their brain structure, then we can have a proper approach in the future for the live specimen. One major challenge is the functional MRI for these guys, but I really want to do this. However, this guy using hemocyanin, so it will provide a very weak signal about oxygen. So we need to have a different approach to increase the signal noise ratio to help us to detect the signal. And another big challenge is because we need to keep them anesthetized and sitting in the seawater and put in the container and into $1 million machine. So I need to convince the manager to allow me to do this very high risk task. So at this stage, we are unable to do the live animal yet. But
Right.
as you can see, we already have a lot of structure detail. So now we can design different approach like electrophysiology or something else to do more functional approach. So we are planning to do metabolism very soon. So we'll measure in different life stages how much oxygen they consume based on their daily task. Thank you.
Thank you. There's another question from Tarun. Wonderful talk. We got to learn a lot. And can you elaborate a bit on how octopus learns cooperative communication with the coral truth?
Communication.
Hmm.
Yeah, we don't really know yet because as I mentioned, this guy very difficult to keep in the lab. So we always have a nightmare. They will try to escape, and some of them have very strong personality. So yeah, so some of them can train, some of them just unable. But we do try to figure out their body pattern for the communication purpose. So we have a lot of imaging recording and try to disentangle what kind body pattern is actually for what kind of communication purpose. So for instance, their courtship display, we already know some species using specific pattern for the communication. Yeah. But for general, we are we aim to use the more social species because they have more interactions, and we'll try to figure out their patterns. One thing I need to emphasize, most of octopus, they are solitary. So they are very rare to encounter another sibling or their neighbor. So it's hard to study how they use those visual communication with their friends or neighbors. Yeah.
Another question from Tarun, but seems like unrelated, but it's interesting. Can octopus increase its camouflage efficiency with age? Say the young versus the more mature individual. Are there any difference or any increasement in terms of camouflage?
Increasement,
In terms of
increasement of what?
camouflage efficiency.
Oh, efficiency. Right. I think in general they are very good because that's how they survive. So, but we do have some spacemen in the captivity because in the aquarium the environment is relatively simple. So somehow they are unable to display very complex patterns. Yeah, but those wild caught, they all of them can do very good camouflage tricks, and then almost immediately will change to match the background. Yeah.
I see. Another question from David. Are the reef octopus species threatened by climate change and reef destruction? Is there any chance that they will be able to adapt to new environment?
Yeah, very good question. Yes. One thing I didn't mention in my talk is octopus actually can use RNA editing to deal with the temperature variation in the environment. So I think a couple years ago, there's a Cell paper. So if you put octopus in a cold water, within 24 hours, their RNA editing capability actually reacts very fast to deal with the cold water. So I guess they might have a capability to deal with the fluctuation of the temperature for some level. However, one thing I also need to emphasize, this guy, especially their embryo, their hatchling, is really affected by the temperature. So if they during the brooding season is too warm, they very likely they will hatch prematurely, so unable to survive. So there are a few cases in Japan and in Korea because the sea temperature increase 1-2 degree, so make the whole spawning aggregation miss out the time. So the population actually collapse because when they hatch, they don't have enough food, so that actually affect the population.
Another question from Catherine. Thanks for the talk. The result about females being able to recognize their own eggs are so cool. Do you hypothesize it's a purely mechanosensory mechanism, or there could be a biochemical signatures?
Yes. Okay. As I mentioned, female keep the chemosensory center intact. However, at this stage, we don't have a single cell recording direct from the sucker. So their sucker is basically can detect all those very fine detailed chemical cue, but we don't have the result yet. That's our ongoing project. We will try to figure out what kind of signal cue is so important for they recognize their brood. Yeah. They will need some recording direct from the cells or from the nerve cord direct from the arm. We haven't done that yet, and we will try to figure out how to do that kind of approach.
I think during your talk, I have another question. During your talk, you mentioned about the brain architectures and behavior that drive behavior. Do you think of another way around, like whether the behavior drive brain architectures?
In some level, yes, we can predict. So based on their behavior, here I only showing my octopus data. We also doing a lot of brain scan in cuttlefish. Yes. So the answer is yes. We can based on their life mode, their behavior, and also well match with their brain design, just as like the octopus.
So based on what you just mentioned now, it seems like you are consider that we can generalize this finding that you present here across other cephalopod species. Is that right?
Well, maybe. Uh, as I mentioned, the diversity in the Indo-Pacific region is so high, so we still know very little about the species in this area. So we now still increase the sample across many different groups. So eventually we will have the mega data set, and then we will have more clear picture what is going on, how they involve in developing different brain structure. Yeah, but it's basically matched with that pattern so far. So I have scanned 35 different species. Now, so far, still within that scheme. So, yeah.
So maybe my last question. Do you have any insights into how nervous circuit connectivity decrease over time in octopus? You mentioned that at certain, it's very always kind of lose on most of the connection, but have you got any survey during the lifetime at any age that it start reducing the connectivity?
We only have the structural evidence. Yeah, we don't know how they really functional across those circuit yet. Yeah, so I guess that probably will need next step. We're doing a lot of simulation and try to figure out if this neural network may be more flexible. Or for instance, when the female brain decay, she still can do a lot of very complex tasks. So they may not need a fully developed circuit, but they can do something else. So maybe they can bypass some broken circuit. Yeah, so that's the new idea. Yeah, that's probably in another 5 years or maybe 10 years to figure out what is going on in their brain, in the
All right.
neuroscience. Yeah.
All right. Thank you. Thank you again, Wen-Sung.